Ever look at a giant tree and wonder how it came from a tiny seed? Or how a cut on your finger just heals itself? Well, it all boils down to this absolutely fundamental process in life, cell division. Today, we are diving right into the grand finale of that process, cytokinesis. That's the incredible moment when one cell finally, officially becomes two.
But that begs a question, right? Why go through all this trouble? Why can't one single cell just keep getting bigger and bigger forever? It seems like it would be a lot simpler. Turns out, though, there are some major physical roadblocks that make that completely impossible. And this problem, well, it's the very reason cells have to divide in the first place.
So, let's get into those roadblocks. As a cell gets bigger, it runs into two huge challenges. It's a situation we can basically call its growth problem.
Okay, this slide really gets to the heart of the problem. As a cell, or in this case these cubes, gets bigger, its volume, all that space on the inside, grows way faster than its surface area, which is the outer membrane. See, a small cell has a huge surface area compared to its volume. That makes it super efficient at moving nutrients in and waste out. A big cell? Nope. It's kind of like trying to supply a massive city using just one tiny two-lane road. It just doesn't work.
So it really boils down to two main issues. First up is DNA overload. Think of the cell's DNA as its central library or command center. If the cell gets too huge, that command center just can't get information out fast enough to run the whole operation. And second, as we just saw, is the material exchange problem. That shrinking surface area to volume ratio means the cell would either starve to death or literally poison itself with its own waste. The only way out? It has to divide.
But here's the thing, a cell can't just decide to split in half whenever it feels like it. Oh no. It takes a ton of careful planning and preparation. Before that split can ever happen, the cell has to go through this incredibly organized prep phase.
And this is it, the cell cycle. And get this, a cell spends most of its life right here, in interphase. This is by far the longest phase, where all that critical prep work gets done. During the G1 part, it grows. Then in the S phase, S stands for synthesis, it meticulously copies every single strand of its DNA. And finally, in G2, it does one last quality check, making sure everything is absolutely perfect before the main event, the division.
So, after all that preparation, the cell enters the first major stage of division, and that's mitosis. Now, this isn't the final split of the whole cell. Not yet. Mitosis is all about the incredibly precise division of the nucleus and all of its precious cargo, making sure each new cell gets a perfect, complete set of genetic blueprints.
You can think of mitosis as this beautiful four-step dance. First, in prophase, the DNA coils up into those classic chromosome shapes we all recognize. Then comes metaphase, where they all line up perfectly down the middle. In anaphase, they get pulled apart to opposite sides of the cell. And finally, in telophase, two brand new nuclei form around the chromosomes at each end. It's this elegant choreography that perfectly sets the stage for the grand finale.
So all of that intense preparation, all that careful sorting of DNA, it has all been leading up to this exact moment. Now we finally get to the great divide, the actual physical splitting of the cell, a process we call cytokinesis.
So what is cytokinesis exactly? Well, if mitosis was all about dividing the blueprints in the main office, the nucleus, then cytokinesis is about splitting the entire factory floor, the cytoplasm, and all the machinery in it into two separate, fully functional factories. It's that final pinch that creates two new cells from one.
Okay, this is where things get really clever. You see, not all cells are built the same way. An animal cell is kind of soft and squishy, but a plant cell is stuck inside a rigid, boxy cell wall. So over millions of years, life came up with two totally different but equally brilliant ways to solve the exact same problem.
And this just nails the core difference, right? Animal cells, since they don't have that rigid wall, they just pinch inward. But plant cells, with those stiff walls, they can't do that. So instead, they have to build a brand new wall right down the middle, from the inside out.
Let's start with the animal cell. See that groove that's forming? That's called a cleavage furrow. And the analogy here is just perfect. It is literally like pulling the drawstring on a hoodie, just cinching it tighter and tighter until it closes all the way. So how does it work? A ring of protein fibers, called a contractile ring, forms right under the cell membrane. Then that ring just tightens and tightens and tightens, pulling the membrane inward with it. That creates the cleavage furrow.
And it just keeps going until, pop, the cell pinches off into two completely separate cells.
All right, now for the plant cell. That super rigid cell wall makes pinching totally impossible. I mean, it'd be like trying to pinch a cardboard box in half. It's just not going to happen. So the plant cell has to take more of a construction-based approach.
Here's the play-by-play. These tiny little sacs called vesicles, which are packed with all the raw materials for a cell wall, they all travel to the middle of the cell. They start to fuse together, forming something called a cell plate. This plate then grows from the center outwards until it connects with the existing cell walls on both sides. And just like that, it's built a new wall and divided the cell in two. Pretty amazing. So after all of that pinching and all of that building, what's the end result? What is this incredibly complex and beautiful process actually made? Well, let's meet the new arrivals. And there they are, two daughter cells. Now, the most important thing to remember here is that they are genetically identical to each other and to the parent cell they came from. They're also smaller, which, hey, solves that whole surface area to volume problem we started with. And now they're all set to grow and, eventually, start this whole cycle all over again.
I mean, it's an absolutely incredible mechanism. But why? What's the big picture purpose of all this dividing? Well, the answer really depends on what kind of organism you are.
You see, for a single-celled organism like this amoeba, this whole process, mitosis followed by cytokinesis, that is reproduction. It's how one parent organism simply divides to create two brand new, totally identical individuals.
But for big, multi-celled organisms like us, it's a completely different story. We use this process for pretty much everything else. It's how we grow from a single fertilized egg into a complex human being. It's how we repair damaged tissue when we get a cut. It is, quite literally, the engine of growth and maintenance for our entire lives.
The precision of cell division is what allows for growth, for healing, really for life as we know it. It is a fundamental process that is kept under incredibly tight control, which leads to one final, really important question. If this process is so vital, what happens when the cell loses control?